So, okay, look, I'm really thrilled to be here. Ian, thanks for including me in this. I was here 8 years ago, and I can't believe it was 8 years ago. It's amazing to be back. I mean, I love it here, and being part of this family is absolutely brilliant. So, thanks for that. I've got some things to say at the end as well, so I'll save them to the end. Look, I didn't know what to talk about today. Last 8 years ago, I gave what I think is sort of a hardcore synthetic chemistry talk, and Ian saw the talk I'm going to give today, and he said, "What happened to you? What you doing?" And so I thought I'd better explain myself here. Before I sort of committed to this, I thought, what would I talk about? I joined Janssen 4 years ago after 25 years at Merck, and so I'm a newcomer there, right? And so I find it a little awkward to talk about everyone else's science when I really haven't been part of it for that long. It's a little tricky, but I thought let me hang on until the last possible minute, because there's something really, really exciting going on in our company right now in the area of multiple myeloma. I wanted our scientists to get out and talk about it first before I did, so I was sort of holding on, and fortunately some of our team have been out and about talking about this now, so I feel comfortable doing this. I also wanted to talk about this because it's probably the most exciting thing that I've been involved in 30 years in pharma. And hopefully, if I get to the end of this on time, I'll show you why. So, as I said, I joined Janssen 4 years ago, and I joined this journey around how are we going to take on and defeat this nasty disease called multiple myeloma. The company had been involved in that for 20 years prior to me even knowing they were doing this, and I joined pretty late on. And it's been an absolutely spectacular ride, and so I want to share some of that with you today. But I want to start with a slide that talks about how we think about taking on disease generally. This is a really, really simplistic view of what disease is. How do I change this? Here we go. All right. There you go. Really simplistic view of disease, okay? So basically this is Rich Tillyer's version of what actually happens in disease, and it's abnormal cell function that is the result of abnormal or missing protein function. So therapeutic interventions restore protein function, which restores cell function and gets you back to where you should be. That's pretty simple, pretty high level. It's actually a lot more complicated than that. But let's talk about what I mean by therapeutic interventions, because today we have the ability to intervene in disease biology in so many ways. It's unbelievable. Relative to like 20 years ago, we can do things today you couldn't have dreamt about even 3-4 years ago. So let's go through them, because there's lots of sort of molecular sizes and shapes that you can engage to go after disease biology. So small molecules, I love small molecules, right? They interact directly with proteins. You can turn a protein on, you can turn it down, you can correct its structure, you can activate, you can make more, you can make less, you can degrade, you can do all kinds of things with small molecules that, again, we're just starting to figure out now. Things like splicing of RNA, right? You can interfere with that with small molecule drug today. So there's all kinds of things you can do with small molecules, and if anyone says small molecules are dead, don't believe them, right? We're actually on a big renaissance here. Small molecule drugs are great. Antibodies are pretty interesting too. You're largely restricted to cell surface and extracellular targets there, but there's a lot you can do, and one of the things I'll show you today is how we use antibodies to label cancer cells for destruction. Okay, that's a really, really important use of antibody therapeutics. Small molecules and antibodies is what most people think about, but you need to go well beyond that if you're going to intervene broadly in disease, because those modalities don't work in every instance, and you never know which one's going to work best. So today you need access to RNA therapeutics. Okay? That could be RNAi, which shuts down protein synthesis. It could be mRNA, which amplifies protein synthesis and adds protein function. You need access to gene therapy, which is replacing a bit of missing DNA or malfunction in DNA into the nucleus to restore protein function. You need access to that technology today if you're going to intervene broadly in disease. And then there's cell therapy, which is the entire cell is actually the drug. And you can now engineer cells to go after, in particular, cancer. And we're going to talk about that today. So in our group, what we've done is we've assembled the entire suite of molecular sizes and shapes. It's a fantastic group. And the reason we do that is because we don't know which ones are going to work, we don't know which ones are going to be the best, but we need to go after disease broadly, and you need access to the entire tool set to do that. So the multiple myeloma story is an example of just that. So let's get into multiple myeloma. What is it? It's a disease of the plasma cells. Plasma cells reside in the bone marrow, and they make all your antibodies for immunity. Pretty important cells. Now, the thing about multiple myeloma is it's a progressive cancer, and it takes time to develop. And you know, there's about 200,000 patients a year that are diagnosed globally, so it's a pretty rare disease. Okay, but it's one of these things that it will creep up on you. And one day you show up in a doctor's office, you have an infection, you may have a bone fracture, you may have renal impairment. Okay, but you show up, and then the diagnosis is from a blood sample or a urine sample where you start seeing these funny proteins. These are mixtures of light chains, misformed antibodies, lots of them. And that's when you're diagnosed with multiple myeloma. Okay, it's a classic telltale sign. When you're diagnosed with multiple myeloma, the survival rate is about 5 years for about 50%. Okay, so 50% of patients, once they're diagnosed, diagnosis is normally in your 60s, 5-year survival rate. Okay, until recently. Okay, so how's it treated? Well, let's talk about how it's formed first, and then we'll talk about how it's treated. So it is a slow progressive disease. It's a result of multiple somatic mutations in the plasma cell that build up over time. The first stage of this is something called monogammopathy of uncertain significance, which means you've got something going on in your blood, and you're making these funny protons, and we're not really sure what they're doing, but it's actually a precancerous state. Okay, and that occurs after the first round of somatic mutations. You just accumulate these over time. Okay, then you get a second hit, and the second hit generally is in a gene that it suffers a mutation in a gene that is highly associated and a driver of cell proliferation. You get a mutation there on top of a whole bunch of somatic mutations, you get multiple myeloma. You show up in the doctor's office, you have these weird proteins that are being formed, your plasma cells are crowded out by malignant cells, okay? And you're generally in pretty bad shape at that point. So, how are you treated? If you're healthy enough at that point, you can undergo a bone marrow transplant. And a number of patients do that. And then straight after the bone marrow transplant, you go into rounds and rounds of maintenance therapy. If you get to 5 years and you're still alive, generally half of the population at that point will be absolutely refractory to any treatment, okay, and out of options. Then it's a downhill slide to death. Okay, this is a pretty significant disease, and it does progress quite aggressively. Okay, how do you treat it? Let's go through the current treatment options. Small molecules. Let's start right there. And if you look at that little structure, it does have a boron in it. So, and this is a proteasome inhibitor. Now look, what's going on here? Cancer cells, especially myeloma cells, are rapidly turning over protein. Okay, and the proteasome is going crazy. Now the proteasome, what it is it's like the garbage disposal mechanism of the cell. All right, so the proteasome takes misformed proteins, excess proteins, aggregates, and processes them back to amino acids and sort of recycles. Okay, this is a very important piece of machinery, and it's going crazy in a cancer cell. You can inhibit the proteasome in cancer cells, and because they're very sensitive to this, because it's highly amplified in the cancer setting, you can actually turn the cancer cell off, and you can actually kill it. And in fact, what happens is you end up stabilizing proteins like p53, anti-apoptotic proteins in addition to that. And essentially what happens there is you flip the cell into cell death. All right? And so that's a good way of addressing multiple myeloma. And you can do that without killing too many healthy cells because you've got this difference in sensitivity. The first proteasome inhibitor was introduced by Janssen 20 years ago in partnership with Millennium Pharmaceuticals. There have been a number since. Okay? But these are widely used. Let's go next, molecular glues. Now, revlimid, you've probably heard of revlimid. It's a pretty interesting compound. Was found to treat myeloma pretty effectively. 14 years after it came to market, after Celgene brought this to market, the mechanism was identified for this. Revlimid is a molecular glue. It basically brings proteins, specific proteins, into the E3 ligase ubiquitination system, which basically tags the proteins for destruction. Okay? Now, there are two transcription regulators that revlimid targets and brings in for destruction. These are called Icaros and Aiolos. Okay? Now, what's important about those two transcription regulators, they do actually regulate the transcription of lots of proteins, but there's one in particular called IRF4. Multiple myeloma cells are addicted to IRF4. If you can shut down IRF4 in a multiple myeloma cell, you'll stop it in its tracks, and that's exactly what revlimid does. So this is a molecular glue that degrades a transcription regulator called Aiolos and Icaros, which then has a downstream effect in shutting down synthesis of IRF4. Okay, and that then halts the cancer. Okay, biologics. There are two I want to talk about here that are really important. There's one which is an anti-CD38 antibody, and basically what you're doing here, this is one on the left, this is a compound that was introduced by Janssen about 10 years ago, and what this does, it targets multiple myeloma cells because they express CD38. Okay? Other cells express CD38 as well, but they're highly, highly expressed on multiple myeloma cells, so you can get some differential activity here. And basically what this antibody does is it labels the cell, and through the Fc portion of the antibody brings in the immune system, brings in T cells to kill the myeloma cell, it brings in complement, it also brings in macrophages as well to engulf and phagocytose the cancer cell. Multiple mechanisms of action here. One other thing we recently discovered with this compound is that it also takes out a population of highly immunosuppressive regulatory T cells that express CD38. We didn't know this population existed until recently. But this is really, really important. These are highly, highly immunosuppressive. If you can take them out, you can get T cell infiltration and then tumor killing. So, pretty simple, right? You've got this target on the tumor cell. It's not on other cells. You bring in an antibody. It brings in the immune system, kills the cell. There's another drug that was invented by GSK, and this uses another tumor ligand, a tumor-associated antigen called BCMA. That's the B-cell maturation antigen. This is expressed really well on malignant myeloma cells, okay? And it's actually not expressed a lot on other cells, so you can imagine a pretty big therapeutic index here if you can target BCMA expressing myeloma cells with an antibody. What GSK did was pretty good. They have this beautiful antibody, right, that targets the myeloma cell, and it's attached through a linker to a toxin. The antibody was optimized to bind to the BCMA on the tumor cell and then to rapidly internalize, dragging the toxin in, which kills the cell. It's like a little nuclear bomb for the cell. Works really well. Now what's interesting about this molecule is that it actually is pretty aggressive, and it causes pretty significant what we call antigenic or immunogenic cell death, which means that as the cell is exploding, it's presenting antigens, which then get taken up by antigen-presenting cells and bring in the T cells to kill the tumor. So this is a very, very powerful mechanism. Okay, so where are we then? These are pretty much what's used today. Over the last 20 years, we've increased the 5-year survival rate from, it was around about 30% to around about 70%. Okay, it's 20 years of work. Okay, 4 really, really important mechanisms. Clearly, though, that's not enough. Okay, we need to do better than this. So, what are we doing now? Okay, so let's go back to this concept of binding an antibody to a tumor selectively and pulling in the immune system. Let's go back to that concept. So what we've done now at Janssen is we've invented a bispecific antibody called teclistamab. Okay, and I'll go through this in a little bit of detail in a second. But basically what it does is it binds the myeloma cell through BCMA, the same tumor target that the GSK drug just used, okay? And it also binds a T cell through CD3. CD3 is a portion of the T cell receptor. It's very important for T cell activation. So what happens is you're essentially dragging together a T cell and a tumor cell, and you're activating the T cell to kill the tumor cell through binding to CD3. This is a really, really powerful mechanism, so much more powerful than anything else to date as far as killing tumor cells. So, but you have to optimize these things, and it took actually quite a long time to get this right. You have to get it right because you can either exhaust the T cells if you go too hard on them, or you can actually, if you don't get the binding affinity right on the tumor cell, you don't get enough tumor killing, right? So you've got to get the relative affinities of these two binding arms exactly right. There's a difference in about 100-fold in binding affinity, and essentially what we did here is we designed the antibody to bind very tightly to the tumor. Okay, so this goes straight to the tumor. It's distributed immediately to the myeloma cells. And then it waits on the tumor cell and grabs a T cell as it comes past. Okay, and that's a low affinity interaction. And the low affinity is really important here so that you don't exhaust the T cell, and also that the T cell is only activated by the antibody that's bound to the tumor. You don't want T cells floating around fully activated. Right? Only should be activated when it's bound to the tumor. So this is a really, really interesting molecule, and in fact I'll talk about the phase 3 data in a minute. But in fact, let's talk about it now. You know, we just had the submission for this. We're waiting for approval from the FDA. But we submitted and got breakthrough status for this based on this clinical data. Now, what you'll see is about a 62% response rate. And you think, well, okay, 62% is good. But let's recognize that these patients are all refractory to current treatment. These patients have no alternative at this point. They've all been heavily, heavily triple treated. It's impossible to treat these patients. So the fact that we're getting 62% responses here is absolutely spectacular, and that was what earned us the breakthrough status. Now, what's really interesting is we're about a year in. I mean, this is a fast-moving program. We've had now, most of these patients are in either remission or in stable disease after a year. Okay, so we're doing pretty well here. We're the hardest to treat patients. Let's just talk a little bit about chemistry here. I really like this thing. This is if you look at the diagram there, the way this antibody is made is we have two separate antibodies that go into this. Okay, one of them is an antibody that has two binding arms for BCMA. The other one has two binding arms for CD3. And the idea is you've got to mix them, right, to make a heterodimer. So the way you do that is, if you look in the FC region there, it's not so clear on the slide, but if you look at the FC region, we've actually put a couple of mutations there that are really important, because what we're going to do is we're going to break those molecules in half. We break a disulfide bond, okay, so it's a simple disulfide reduction, okay, and then we allow the antibodies to reform. And what drives the reformation to completely the heterodimer are those mutations that we put in the FC region. It really, really works well. We're scaling this up now and ready to launch this product, and it's working pretty well. We'll make tons of this a year. So, okay, great drug, and we're really, really excited about this. We're hoping to get approval really soon. This is going to make a big difference to patients that don't respond in any other way. But we want to do better than that, okay? And so let's go back to the concept of the T cell, right? So what we've done here is we've pulled in a T cell to the tumor with a bispecific antibody. If you like, we're helping the kinetics here. Another way to think about going after this problem is, how about if we could take the person's T cells and actually engineer them in a way so they were really, really potent tumor killers, right? That's a technology you've probably all heard of called CAR-T. We got into this space because we knew that there are lots of patients that don't have healthy T cells. And you can put a redirector in there, but if you don't have a T cell to redirect, it's not going to work. And so we went after the the ultimate solution here, which is how do you actually take someone's T cells, re-engineer them, put them back in, and have them go after the tumor. So that's what we're doing. Let's just go through that now. So we actually just had approval for this. This is our first cell therapy product at Janssen. It's called Cilta-cel. And this is a pretty amazing product. I'll show you some data on it in a minute. But basically what you're doing is you're introducing into the person's T cells an engineered T cell, an engineered receptor. Okay? That receptor, there's a cartoon of it on the right. It has an extracellular domain, which is these two VHHs, two heavy chain antibodies, if you like, that both bind to BCMA. It's really important that you have two. Okay? The intracellular signaling domain is made up of a CD3, which, as I said, is important in T cell activation, and a co-stimulatory domain called 41BB. Now, I didn't pull this out of thin air. This is the result of sort of 20 years of research across many, many places, all right? But take it from me that when the T cell is engineered this way, re-administered to the human, the binding domain binds to BCMA on the tumor. That causes an activation of the T cell to secrete cytokines to kill the tumor. Let's show you the data. So this is pretty spectacular. I've never seen anything like this, to be honest. We have 98% responses of in this trial. Every patient was out of options. These folks were literally weeks away from dying, and almost all of them got up, walked away, and are leading healthy lives. Okay, it's pretty amazing. And these responses are actually growing with time, meaning that they're deepening with time. We're now 2 years in. Patients are still doing well. And to be honest, we have difficulty tracking these patients down. We try and find where they are, and they're all living healthy lives. And they don't want to talk to us anymore because they've got their lives to live. But the fact is they're doing really, really well. Okay, so we have approval for this product. It's now in full-scale manufacture, and we are trying to expand the utility of this. So, last slide. I think I've shown you that you can use all of these modalities to treat a cancer like this. Small molecules, proteins, ADCs, multispecifics, and now cell therapy. And what I'm going to do for the next 5 years of my career is I'm going to figure out how Cilta-cel works. Well, I'm not going to. My team's going to figure that out. And because you get efficacy like that, and you have to figure out what's driving that, and then we have to figure out exactly what are the components of because these cells are big mixtures, right? We have to figure out what are the active components there, and then we're going to try and figure out how to expand the utility beyond multiple myeloma to other tumor types, especially into solid tumors. So that's what I'm going to be doing for the next 5 years. Really looking forward to it. Last slide here, I want to thank everyone that's been working on this program all the way for the last 20 odd years, especially my colleagues at Janssen. Other companies in the industry also are making massive impact here. And lastly, I want to thank Ian, because I have, I think, the best job in the world. And, I get to work with people like this that are doing this. And I wouldn't have had that, Ian, if it wasn't for you. Because most of you guys don't know this, but I was in a bit of a rush coming out of my graduate studies. And I was locked in to go somewhere to go and do a postdoc, and it all fell through at the last minute. And Ian sort of plucked me out of thin air and says, come into my team and come to Cambridge and come be a postdoc in my group. And, sometimes I think, Ian, if that hadn't happened, I wouldn't be in a job like this. So it's all down to you. So thank you very much.